Okay, reading. Starting at potassium is subject to competing forces. Potassium is positively charged and the inside of the cell is negatively charged. So the electrical gradient tends to pull potassium in. However, potassium is more concentrated inside the cell than outside, so the concentration gradient tends to drive it out. For an analogy, imagine a number of women inside a room. Men can enter the room or leave through a narrow door. They are attracted to the women, but when the men get too crowded, some of them leave. The concentration gradient counteracts the attraction. If the potassium channels were wide open, potassium would have a small net flow out of the cell. That is, the electrical gradient and concentration gradient for potassium are almost in balance, but not quite. The sodium potassium pump continues pulling potassium into the cell, counteracting the ions that leak out. The cell has negative ions too. Negatively charged proteins inside the cell sustain the membrane's polarization. Chloride ions, being negatively charged, are mainly outside the cell. When the membrane is at rest, the concentration gradient and electrical gradient balance, so opening the chloride channels would produce little effect. However, chloride does have a net flow when the membrane's polarization changes. Why a resting potential? The body invests much energy to operate the sodium potassium pump, which maintains the resting potential. Why is it worth so much energy? The resting potential prepares the neuron to respond rapidly. As we shall see in the next section, excitation of the neuron opens channels that allow sodium to enter the cell rapidly. Because the membrane did its work in advance by maintaining the concentration gradient for sodium, the cell is prepared to respond vigorously to a stimulus. Compare the resting potential of a neuron to a poised bow and arrow. An archer who pulls the bow in advance is ready to fire at the appropriate moment. The neuron uses the same strategy. The resting potential remains stable until the neuron is stimulated. Ordinarily, stimulation of the neuron takes place at synapses, which we consider in Chapter 2 in the laboratory. It is also possible to stimulate a neuron by inserting an electrode into it and applying current. The action potential. Messages sent by axons are called action potentials. To understand action potentials, let's begin by considering what happens when the resting potential is disturbed. We can measure a neuron's potential with a microelectrode, as shown in figure 1.13. When an axon's membrane is at rest, the recordings show a negative potential inside the axon. If we now use a different electrode to apply a negative charge, we can further increase the negative charge inside the neuron. The charge is called hyperpolarization, which means increased polarization. When the stimulation ends, the charge returns to its original resting level. The recording looks like this, an image. Now let's apply a current to depolarize the neuron, that is, reduced its polarization towards zero. If we apply a small depolarizing current, we get a result like this image. With a slightly stronger depolarizing current, the potential rises slightly higher, but again returns to the resting level as soon as the stimulation ceases. Image. Now let's apply a still stronger current. Stimulation beyond the threshold of excitation produces a massive depolarization of the membrane. When the potential reaches the threshold, the membrane opens its sodium channels and lets sodium ions flow into the cell. The potential shoots up far beyond the strength that the stimulus provided. Any sub-threshold stimulation produces a small response that quickly decays any stimulation beyond the threshold image. Regardless of how far beyond, produces a big response like the one shown, known as the action potential. The peak of the action potential, shown as plus 30 millivolts, in this illustration varies from one axon to another. Stop and check. The all or none law. Note that any depolarization that reaches or passes the threshold produces an action potential. For a given neuron, all action potentials are approximately equal in amplitude, intensity, and velocity. That is, the intensity of the stimulus cannot cause a neuron to produce a bigger or smaller action potential or a faster or slower one. Slight variations can occur at random, but not because of the stimulus. More properly stated, the all or none law is the amplitude and velocity of an action potential are independent of the intensity of the stimulus that initiated it. More properly stated, the all or none law is that the amplitude and velocity of an action potential are independent of intensity of the stimulus that initiated it, provided that the stimulus reaches the threshold. By analogy, imagine flushing a toilet. If you have to make a press of at least a certain strength, the threshold, but pressing harder does not make the toilet flush faster or more vigorously. Similarly, when you flick the switch to turn on the lights in your room, flicking the switch harder would not make the lights brighter. Okay. Although the amplitude, velocity, and shape of action potentials are consistent over time for a given axon, they vary from one neuron to another. Thicker axons convey action potentials at greater velocities. Thicker axons can also convey more action potentials per second. The all or none law puts constraints on how an axon can send a message to signal the difference between a weak stimulus and a strong stimulus. The axon cannot send bigger or faster action potentials. All it can change is the timing. By analogy, you might send signals to someone by flashing the lights in your room on and off, varying the speed or rhythm of flashing. Flash, flash, dot, 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 long pause, dot, 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 flash, flash. Might mean something different from flash, dot, 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 pause, dot, 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 flash, dot, 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 pause, dot, 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 flash, dot, 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 etc. The nervous system uses both kinds of coding. For example, a taste axon shows one rhythm of responses for sweet tastes and a different rhythm for bitter tastes. The molecular basis of action potential. The chemical events behind the action potential may seem complex, but they make sense if you remember three principles. At the start, sodium ions are mostly outside the neuron, and potassium ions are mostly inside. When the membrane is depolarized, sodium and potassium channels in the membrane open. At the peak of the action potential, the sodium channels close. A neuron's membrane contains cylindrical proteins like the ones in figure 1.12. Opening one of these proteins allows a particular type of ion to cross the membrane. Which ion crosses depends on the size and shape of the opening. A protein that allows sodium to cross is called a sodium channel, or gate, and one that allows potassium to cross is a potassium channel. The axon channels regulating sodium and potassium are voltage-gated channels. That is, their permeability depends on the voltage difference across the membrane. At the resting potential, the sodium channels are fully closed, and the potassium channels are almost closed, allowing only a little flow of potassium. As the membrane becomes depolarized, both the sodium and the potassium channels begin to open, allowing freer flow. At first, opening the potassium channels makes little difference because the concentration gradient and electrical gradient are almost in balance anyway. However, opening the sodium channels makes a big difference because both the electrical gradient and the concentration gradient tend to drive sodium ions into the neuron. When the depolarization reaches the threshold of the membrane, the sodium channels open wide enough for sodium to flow freely, driven by both the concentration gradient and the electrical gradient. The sodium ions enter the cell rapidly until the electrical potential across the membrane passes beyond zero to a reversed polarity, as showing in the following image. Of the total number of sodium ions near the axon, less than 1% cross the membrane during an action potential. Even at the peak of the action potential, sodium ions continue to be far more concentrated outside the neuron than inside. Because of the persisting concentration gradient, sodium ions still tend to diffuse into the cell. However, at the peak of the action potential, the sodium gates snap shut. Then what happens? Remember that depolarizing the membrane also opens potassium channels. At first, opening those channels made little difference. However, after so many sodium ions have crossed the membrane, the inside of the cell has a slight positive charge instead of its usual negative charge. At this point, both the concentration gradient and the electrical gradient drive potassium ions out of the cell. As they flow out of the axon, they carry out with them a positive charge. Because the potassium channels remain open after the sodium channels close, enough potassium ions leave to drive the membrane beyond its usual resting level to a temporary hyperpolarization. Figure 1.16 summarizes the key movements of ions during an action potential. At the end of this process, the membrane has returned to its resting potential, but the inside of the neuron has slightly more sodium ions and slightly fewer potassium ions than before. Eventually, the sodium-potassium pump restores the original distribution of ions. But that process takes time. After an unusually rapid series of action potentials, the pump cannot keep up with the action, and sodium accumulates within the action. Excessive buildup of sodium can be toxic to a cell. Excessive stimulation occurs only under abnormal conditions. However, such as during a stroke or after the use of certain drugs, don't worry that thinking too hard will explode your brain cells. Action potentials require the flow of sodium and potassium. Local anesthetic drugs, such as Novocaine and xylocaine, attach to the sodium channels of the membrane, preventing sodium ions from entering. When a dentist administers Novocaine before drilling into one of your teeth, your receptors are screaming, pain, 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 but the axons cannot transmit the message to your brain, and so you don't feel it. Propagation of the action potential. Up to this point, we have considered how the action potential occurs at one point on the axon. Now let us consider how it moves down the axon. Remember, it is important for axons to convey impulses without any loss of strength or over distance. During an action potential, sodium ions enter a point on the axon. Temporarily, the spot is positively charged in comparison with neighboring areas along the axon. The positive ions flow within the axon to neighboring regions. The positive charges slightly depolarize the next area of the membrane, causing it to reach its threshold and open its voltage-gated sodium channels. Then the membrane regenerates the action potential at that point. In this manner, the action potential travels along the axon, as in figure 1.17. The term propagation of the action potential describes the transmission of an action potential down an axon. The propagation of an animal species is the production of offspring. In a sense, the action potential gives birth to a new action potential at each point along the axon. Images. An action potential always starts in an axon and propagates without loss from start to finish. However, at its start, it back propagates into cell body and dendrites. The cell body and dendrites do not conduct action potentials in the same way that axons do, but they passively register the electrical event that started in the nearby axon. This backpropagation is important. When an action potential backpropagates into a dendrite, the dendrite becomes more susceptible to the structural changes responsible for learning. Let's review the action potential. When an area of axon membrane reaches its threshold of excitation, sodium channels and potassium channels open. At first, the opening of potassium channels produces little effect. Opening sodium channels lets sodium ions rush into the axon. Positive charge flows down the axon and opens voltage-gated sodium channels at the next point. At the peak of the action potential, the sodium gates snap shut. They remain closed for the next millisecond or so, despite the depolarization of the membrane. Because voltage-gated potassium channels remain open, potassium ions flow out of the axon, returning the membrane toward its original depolarization. A few milliseconds later, the voltage-dependent potassium channels close. All of this may seem like a lot to memorize, but it is not. Everything flows logically from the facts that voltage-gated sodium and potassium channels open when the membrane is depolarized, and that sodium channels snap shut at the peak of the action potential. The myelin sheath and saltatory conduction. In the thinnest axons, action potentials travel at a velocity of less than 1 meter per second. Increasing the diameter brings conduction velocity up to about 10 meters a second. At that speed, an impulse along an axon between a giraffe's spinal cord and its foot takes about a half a second. To increase the speed still more, vertebrate axons evolved a special mechanisms, sheaths of myelin, an insulating material composed of fats and proteins. Consider the following analogy. Suppose your job is to take written messages over a long distance without using any mechanical device. Taking each message and running with it would be reliable but slow. Take the propagation of an action potential along an unmyelinated axon. If you tied each message to a ball and threw it, you could increase the speed. But your throws would not travel far enough. The best solution would be to station people at moderate distances along the route and throw the message-bearing ball from person to person until it reaches its destination. The same principle applies to myelinated axons. Those covered with a myelin sheath, myelinated axons, found only in vertebrates, are covered with layers of fats and proteins. The myelin sheath is interrupted periodically by short sections of axon, called nodes of Ranvier, each one about 1 micrometer wide, as shown in figure 1.18. In myelinated axons, the action potential starts at the first node of Ranvier. Suppose an action potential occurs at the first myelin segment. The action potential cannot regenerate along the membrane between nodes because sodium channels are virtually absent between nodes. After an action potential occurs at a node, sodium ions enter the axon and diffuse, pushing a chain of positive charge along the axon to the next node, where they regenerate the action potential. This flow of charge moves considerably faster than the regeneration of an action potential at each point along the axon. The jumping of action potentials from node to node is referred to as saltatory conduction, from the Latin word saltare, meaning to jump. The same root shows up in the word somersault. In addition to providing rapid conduction of impulses, saltatory conduction conserves energy instead of emitting sodium ions at every point along the axon and then having to pump them out via the sodium potassium pump. A myelinated axon emits sodium only at its nodes. In multiple sclerosis, the immune system attacks myelin sheaths. An axon that never had a myelin sheath conducts impulses slowly but steadily, but an axon that has lost its myelin is not the same because it lacks sodium channels where the myelin used to be. Consequently, most action potentials die out between one node and the next. People with multiple sclerosis suffer a variety of impairments, ranging from visual impairments to poor muscle coordination.